Hi everyone, my name's Ellen. I'm from the University of Melbourne in Australia, and so I'll be talking about gene drives, which is a sort of new and up and coming technology that might be on some people's radar, and how we might use it to manage or even eradicate particular invasive species in Australia. But before I get into it, I'd like to begin by acknowledging the traditional owners of the land on which this research is taking place, which is the people of the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present. So thinking about what invasive species have meant for Australia, we've spent almost $400 billion on trying to manage invasive species since the 1960s, but the author of this particular paper, which came out, I think it was last year, estimates that this is much, much higher. So cats have been the most expensive by far. The efforts to remove cats haven't been the most successful, but we've certainly spent the most trying to manage them. Management techniques differ greatly depending on the species that you're trying to target, which is obviously not news to many people here. When we're thinking about vertebrate species, which is what I'll be predominantly speaking about today, we sort of rely on a lot of the traditional techniques, which is your baiting, trapping, poisoning, all these sorts of things. But they're really resource intensive, as we've sort of heard already. They're, depending on where you're working, they're sometimes not very effective. So we have problems on our mainland where we might pull out as many animals as we can, but we just have reinvasion from neighboring areas. So on places like offshore islands or fenced reserves, these can be fantastic, but we need something for a large scale control of certain species. So some of the emerging techniques that are coming through, we've got some of the biological controls, which is often your viruses. So the most famous, I guess, is probably the rabbit viral controls of I think it was the 1930s and 40s, which was very successful at removing rabbits, but now they're sort of working alongside trying to optimize. A big issue was resistance. The rabbits became resistant, and so they've had multiple strains come through. We've had physical technologies come through, like the Felixa grooming trap, which is still in development. It basically sprays like a sticky substance onto cats when they walk past, and then they ingest that poison when they groom themselves. We have lots of sort of new aggregating technologies, like for deer is a big one. There's immunocontraception, which is where you basically manipulate the host's immune system so that they basically destroy their own reproductive potential. But the one I want to talk about, which has sort of burst onto the scene in the last, I suppose, 5 to 10 years, is gene drives. Gene drives, I'll go into a little bit about what they are exactly, but it's the mixing of gene drives with the CRISPR-Cas9 editing system that really is showing a lot of promise for invasive species management. So what are gene drives? Well, gene drives are actually a naturally occurring phenomenon. They relate to basically the transmission of a gene or a gene variant from parent to offspring, so what we call vertical transmission. What it's characterized by is an increase in the frequency of that gene, even if it confers absolutely no advantage to that animal. So if you have something like a normal inheritance, or what we might call Mendelian inheritance, you may say that we have, say, 50-50 chance of inheriting a gene from a parent. So if you look at this sort of scenario on the left here, if we're thinking about a red gene under 50-50 normal inheritance, by that F3 generation, we've only got 1 out of the 8 individuals carrying that gene. A gene drive will copy itself into every single offspring, and then every offspring of that offspring, until after a certain amount of time, every single individual in that population will carry the gene drive. So how it works is kind of the most important sort of feature when we're thinking about what we might use it for, because gene drives can be coupled with other things to do to achieve a lot of different outcomes, basically. So if we were to start with a diploid organism, so let's imagine that this is an egg and a sperm that have just met, we've got a fertilized diploid organism, we introduce a CRISPR-Cas9 system. So this will have, I'll use the pointer if it works. Nope, hang on, that one. Yeah, so we'll have this CRISPR-Cas9 gene drive construct here. So if this is your diploid organism here, these black regions represent regions of homology to a particular gene location in the genome. The other things that we introduce alongside these regions of homology is the CRISPR-Cas9 enzyme, the modified DNA, so what we actually want to change that particular gene, and also guide RNA. So what happens when this gets introduced into this system is that the CRISPR-Cas9 and the guide RNA will do their thing. They will go and cut at a particular site that we have told it to cut at. Now, this is the most probably one of the most important parts. What we hope happens is this process called homology-directed repair will basically be favoured in this scenario, where the introduction of the CRISPR-Cas9 modified DNA and guide RNA gets copied over as a form of DNA repair by that particular environment. So this is what we hope happens, and then because we've included alongside this construct the CRISPR-Cas9 and a guide RNA, we've not only given it the gene drive situation, but we've also given it the instructions to keep doing it again and again whenever it encounters. So this is how it copies itself into every single offspring or every single bit of DNA that matches up. So we might use this for lots of reasons. We might use it to reduce a population in the case of invasive species. This might be by, for example, modifying the sex ratio in the population. So one of the best ways we can crash a population is if we make it male biased, so remove as many females as we can. We might make it so that particular female fertility genes are really heavily impacted, so they might have less offspring than they normally would, or none at all. In terms of replacing populations, this might come about in something like disease suppression. So malaria is probably the most common example given right now in terms of using a gene drive, because it's looking like now that we actually won't be able to get rid of malaria unless we replace the mosquito population that transmits malaria with a gene drive mosquito population that can no longer transmit malaria, if that makes sense. So that's one situation where we might drive to replace a population rather than reduce it. We might also try to engineer crops that are resistant to highly humid or drought-resistant conditions to sort of try and adapt towards these changing climate conditions that we have. So obviously I'm going to talk a little bit about more vertebrate species, because these are the big issue for us in Australia at the moment. We have lots of invasive species issues, but these are the ones I'm just choosing to focus on. So this work is being led by Stephen Frankenberg, who is here, and it's called the Piper Lab, and this is our current hit list of species that we're looking at the moment. So we have the European rabbit and the red fox, which are major issues for a number of reasons. And then we also have the European carp, the cane toad, and the smooth newt. The smooth newt is one that surprises people because it only arrived here, well, we only detected its arrival about 10 years ago. So that's a very interesting case study because it's not widely established yet. So the point of the Piper Lab, I guess, is to create multiple different pipelines for multiple different types of animals, and they all benefit, all these pipelines benefit off one another. So the more we develop one for foxes, the more it benefits rabbits, and so on and so on. So the idea is, of course, not to just make gene drives for our established species, but also to be able to respond really, really quickly down the track when we have the new introduction, the new rabbit, the new fox, whether that's a squirrel or I'm not sure what it would be, but have these pipelines ready to go so that when these animals do arrive, we can respond really fast. That's the idea behind it. So this is kind of getting in more the nitty-gritty science of how do we actually go about making a gene drive animal, because I think this is really cool. So I'm going to use foxes as an example for a gene drive, but just remember that I'm using foxes as an example, but that doesn't mean that this couldn't be used for carp or for a cane toad or a rabbit or a cat. So what we go about starting off with is we first take some cells from, in this case, a male fox. We can take skin cells, we can take lots of types of cells, and we're going to try and reprogram those cells back to what's called a stem cell state or an iPSC state. Now, when they're in this state, it means that the cells are far more, amenable. They're kind of more open to genomic changes. So when they're in this state, we're going to introduce something called a LOXP, which is one half of a system called the LOXP recombinase system. And in this situation, we might choose to target a female fertility gene. So maybe this is something like the zona pellucida, which is this protein that surrounds the egg layer. And if it's not there, sperm can't bind, fertilization can't occur. So what we do, rather than trying to put the entire gene drive construct right into this female fertility gene, which is a long, big, chunky thing of DNA, we put these lock sites in. Now, what that means is that anything can go into those lock sites if it's provided with its counterpart, the CRISPR recombinase system. So what we do is we get sort of this gene ready, I guess, to be disrupted down the track by the introduction of whatever we would like, most likely a gene drive construct. So these, the genome of these fox cells have been put back into a stem cell state. They've been modified now, so they have the LOXP sites in this fertility gene. And what happens is we take a fertilized fox egg, so something that's just been fertilized by sperm. We take the nucleus out, which is now called an enucleated egg. So it's just been fertilized, but the nucleus has been taken out. We replace it with the genome from these stem cells that have the LOXP and the fertility gene. And we essentially, hopefully everyone's still with me, have made a clone of this original male that he now has carrying a LOXP sites and the fertility gene. This male is then mated to a wild type female. When this fertilization occurs, this is when we introduce our CRISPR system. So this is the system that's going to allow us to swap in whatever extra DNA or constructs we'd like into that female fertility gene. So this, in this case, would be the gene drive construct, so that allowing every offspring of this resulting male or progeny to continue on with this gene drive pipeline. So hopefully everyone's still with me at this stage. It's a bit of a convoluted process, but and it's complicated as well. We're sort of still in this sort of realm here, but we're hoping that, as we work on multiple species, that this whole process gets much easier. So in terms of thinking about gene drives, there's obviously a lot to think about with something like a gene drive to manage species like this, because it has the potential for complete eradication. I think it's really important to note that invasive species are among the biggest threats we have in Australia. It's the same here in New Zealand. When we think about our environment, our agriculture, and more, I guess, is being talked about more and more is our health as well, that One Health approach. Gene drives have the potential to do a lot of things. They have the potential to be more humane. None of this requires culling or causing any real harm to animals. They shouldn't even know anything's happening. It's highly species specific. So the chances of, it jumping outside of its host is almost near non-existent, because in a place like New Zealand or Australia, all of our wildlife are incredibly unique. A red fox cannot mate with any of our animals, so it can't pass on this gene drive. It's completely restricted to just red foxes. And it's highly effective. It has the potential to be highly effective at actually removing the animals. As we've seen, it's hugely resource intensive to try and remove animals at the scale that we need to do it, and this might be one of the tools that we use for it. And I think the other thing to point out is that gene drives may be appropriate for places outside of Australia as well. So Australia may be able to use gene drives because of our isolated geographic structure of our country. New Zealand is very much the same, and there's a few other options as well. Of course, this sort of stuff takes decades to work on, and whether or not gene drives are ever the one answer, I guess, remains to be seen. We need a lot more work, but I think what we can all agree on is that this problem is only getting worse. I'd rather us all be in a situation in 20 years where we have as many options available to us as possible, and then we can sort of decide on what it might be appropriate. So I'm going to leave it there, and there's so many people to acknowledge, but I really want to point out the Invasive Hub for helping me get here today as well. I really appreciate your help. So thanks very much, and happy to take questions.
Thank you for that. Has anybody got any? Oh, thank you for that. I'm just wondering where we are at in regard to public acceptance of gene drives, and particularly, I know that CSIRO recently conducted, a series of surveys and found that 3/4 of people responding to that survey felt that any interference in nature doesn't end well. How do you think we're going to ever see this technology being used because of that public acceptance?
Yeah, absolutely. So the acceptance rate is something that's being discussed and investigated a lot. So the most recent publication that came out from CSIRO was looking at gene drive acceptance rates in cats. So they went straight to probably the most controversial animal that you would use a gene drive for. They found around, don't quote me, but it was around 84% were in favor of a gene drive for feral cats. And I don't mean strongly supportive, I mean within the somewhat supportive through to strongly supportive. So there were some in that highly disagree category. About 95% of Australians in this study also said that they are in favour of a gene drive to control invasive species, so not specific to cats. So Australians as a whole are very supportive of invasive species control. There's still, exactly as you mentioned, considerable ways to go with public acceptance. So just as important as developing safe and robust science is actual responsible communication and discussion with all parties, because this is not an insignificant thing that we're proposing. But of course, it's really important to remember that this is decades down the tracks and we need as many options as possible. If it turns out it's not the right option, it's not the right option. But I think we get to a stage where we can do this and we engineer as many safeguards as possible along the way and then reassess.
We've had a question pop up online that says that Price and Lindholm 2020 show many possible mechanisms for gene drive resistance. A better drive construct is it causing a drive, the stronger will be the selection on each of those resistance mechanisms, and how will you deal with that?
Yeah, this is a really tricky thing, is that it's actually far easier to make an animal that's resistant to a gene drive than to make a gene drive animal. So this is part of actually what we would incorporate into safety planning for releases or escapes back to locations where the animals are endemic. So what we would also be doing is trying to engineer the gene drives so that the chance of resistance appearing, which means the amount of DNA that's remaining at the particular site we're targeting with this gene drive, is reduced to as little as possible, so that there's less DNA there for changes to arise, which would make the animal resistant. We'd also have to have multiple gene drives ready to go, so and continuous monitoring of when resistant animals pop up as well. So basically planning for all of those occurrences. Yeah.
We got any more?
More questions.
Have you worked out how long a gene drive for foxes would actually take to work? Because I did some work on possums and it was over a century to have any effect because of the generation time.
Yeah, absolutely. So there's a lot of modeling as well going into gene drives. In fact, modeling of gene drives is turning out to be, I think, as big of a field as the actual making of a gene drive. So it very much depends on a lot of things. So the type of gene drive. So something like disruption of female fertility is not going to be as effective as something that completely cuts out all females from the population, and like a sex bias would be far more effective, and that cuts down that time until eradication considerably. So it really depends on the gene drive, depends on the animal, depends on the location, depends on their number of offspring, their generation time, and so on. But it's a really important point. Yeah.
I saw a few more hands.
Hi, this may be a really stupid question, but in malaria mosquitoes, a lot of the discussion around gene drives revolves around the potential ecological impact of eradicating several species from potentially an entire continent. Do you anticipate any sort of negative ecological consequences of eradicating foxes from Australia?
Yeah, so just quickly on the malaria one, I think the idea, although it's not my area, is not to eradicate the mosquito, it's just to replace it with a population that can't transmit malaria anymore. Because exactly right, as soon as you remove a species, you basically opened up this gaping hole in the ecosystem and you don't know what's going to move into that. So the removing invasive species is a really interesting one, because there's all these intricate dynamics. So for example, when the cane toad arrived, it obviously wiped out a lot of our animals, but it also meant that some animals that were endangered went up in their population number, because the animals that the cane toad was knocking out were actually predating on this endangered species, and they increased. So yeah, it's really hard to know, if we get rid of the foxes, are the rabbits just going to go through the roof? Are the cats just going to go through the roof because they're not preying, they don't have competition? So I'd imagine that we'd have to, and this is in decades down the track, once we do have, and it looks like we might have some options for control, I think we'd all need to sit down and come out basically with a long timeline plan of what needs to happen with what species where, because it's just so hard to know. It's just, we don't understand these processes well enough to predict it. Yeah, it's a really good point. Yeah. Thanks.
Do we have you on there?
Yes, I think so.
Oh, excellent. All right, now we hear you.
I don't know if you can see me though.
No, but at least we can hear you.
Okay.
Awesome. Thank you for a really interesting talk. I was just curious, you highlighted with the macrophytes that you found some invasive species and were able to date when they came in. I'm wondering if you noticed anything else, any other interesting species introductions or changes driving some of those other community shifts that you found?
So I wasn't specifically focused on the other than perch, actually. I just brought the invasive macrophytes into the picture because I thought everyone might be interested. So I haven't done a deep dive into all the other invasive species, but definitely now we can kind of put a date to some of these timings. We can potentially look a bit more intricately at some of those changes. But right now we're kind of focused at this high level total community shifts that we can see. Yeah, there are some limitations around taxonomically identifying some of the species within the bigger community groups and stuff like that. But yeah, it would be awesome if we could take a better deep dive, especially in the macrophyte community area. Thank you.